All right, so I'll tell you about, I hope you can hear me okay, um, a project that we recently published. As with many people here, there are many projects and many interesting aspects, but hopefully this is an interesting story. So I'll first tell you, this is a project on ion channels, specifically potassium channels. And I'll briefly give you some background on ion channels in health and disease. The way we got started on this particular project, because we were interested in post-translational modifications, and we were investigating a palmitoylation of one of the KATP channels. And in looking into that, we uncovered some mechanistic insights that when we then looked at the structure, we saw an opportunity for development of ligands that could tell us more about the mechanism and could potentially be drugs. But it's really the mechanism that we're focused on. So I think everyone is familiar with ion channels, fundamental to biology, certainly to medicine, sodium, calcium, and potassium channels. The key thing these are membrane proteins, and as Seva pointed out, this is recent developments have brought membrane proteins much more into the fore. We now have some confidence in the structures, and the algorithms have gotten to the point where once they were unapproachable, now we can use rational design approaches. But they've always been important drug targets, one of the most common targets of clinically used drugs, and there's just an enormous diversity of them. The potassium channels fall into 4 groups. I'll be talking about the KATP channels, or ATP-activated potassium channels. And potassium is high in the cell, and sodium is high outside the cell, and so it creates the electrical potential that is the driver of much of life and cognition. So in terms of the KIR channel, so the ATP sensitive and the ATP sensitive group, there's 4 main families, and I'll be focusing on the ATP sensitive group, but they are similar in structure. They appear to have been divergent from a similar ancestor. And so selectivity of any ligands across this family is probably important. And physiologically, the KATP channels are important in glucose regulation, metabolism, from a pathology point of view, and diabetes. Basically, that when they are triggered, sorry, hold on a second, I'm getting some. When they close, it triggers insulin secretion. And there actually are targets of some diabetes drugs, the sulfonylureas, that block the channels to increase insulin secretion in diabetic states. And more generally, the KATP channels, they're more widely distributed, and they couple energy metabolism inside the cell to membrane effects, excitability of the membrane, or secretory events. So that's important in a variety of tissues, not just in the pancreas. Regulation of blood pressure, secretion in neurons for synaptic transmission, and so on. And in addition, they are targeted by several of the drugs that are involved in hair regrowth. So they're important class of drug targets in medicine and biology, and we became interested in this because we were with a collaborator. We were interested in post-translational modifications, and one of the less investigated post-translational modifications, which is palmitylation, which, like glycosylation, underlies trafficking of proteins, but we have the hypothesis that both palmitylation and glycosylation can have structural effects on the protein, and therefore functional effects. palmitoylation is conferred by the DHHC acetyltransferases, which are juxtamembrane, transmembrane enzymes that palmitylate cysteines within that are near the cytoplasmic interface. And Huaqin Yang was the lead author on this. He noticed that there are conserved cis residues in the M2 helix, which would be at the membrane interface and an appropriate place for palmitylation. And the residue is predicted computationally to be palmitylated. So we were investigating that. It appears, as you see from the sequence alignment in the lower right, that it's only active in some of the KR family members, the KR6, 3, and 2, KR6 and 3 family members, but not in the 2s, 4s, or 7s. And so using click chemistry to try to detect palmitylation, we did some experiments in cells showing that, and I'll talk a little bit about SUR1. It's a companion cofactor of KR6.2. I'll talk about that in a second when we talk about the structure, but they operate as a complex. But it was clear that that KR6.2 is palmitylated. We're detecting it by biotin incorporation, and caveolin is a positive control, cal module, and a negative control here. More importantly, it might be palmitylated, but that might just be a trafficking effect. And so again, our hypothesis was that it may affect the function. And in fact, when we did patch clamp experiments on the cells, turning the palmitylation on and off, what we could see was that palmitylation increases the KATP and specifically KIR6.2 channel current. So it was it increased the open probability of the channel, which was interesting. There aren't that many things that do that. And importantly, we were able to map the palmitylation to cysteine 166 by mutating it and showing that it abolished the palmitylation and also abolished the functional effect of palmitylation. So this created a bit of a mystery, because when we looked at the structure, what I'm showing here is only the KIR channel without its SUR subunit. The cysteine 166 is actually pointing away from the pore, as shown here. And so it wasn't that clear how it is that palmitylation could promote the opening of the channel. And so the effect must be allosteric. So this is where ICM came in. We did some modeling but before I get to that, let's talk a little bit about the structure, some structural features of the potassium channels and their SUR subunits. So there have been cryo-EM structures of these channels, and the potassium channel, which I just showed you here, is actually only a homotetramer at the center of a complex that is found in biology. It's each monomer is in complex with an SUR subunit, and SUR is a 10 transmembrane channel-like protein, but doesn't have a pore and doesn't transmit any ions. Both the KIR channel and the SUR have a nucleotide binding cytoplasmic subdomain that helps at regulation in the overall. And the SUR subunit is essentially an inhibitory subunit of the channel. The channel itself has a series of gates that that control the flow of ions through the channel. A wider gate near the cytoplasmic membrane where the nucleotide binding domain is, and then constricted gates going further up. But this key area at the interface of the cytoplasmic face of the membrane is which is also the interface of the transmembrane helices with this nucleotide binding domain, is kind of like the watershed area of the channel. And the way these channels, at least the way we think they work, is with tilting and rotating of the central helices surrounding the pores, which creates and modifies this series of constrictions at the gates into different shapes, which the constrictions are moved and widened in the open channel and focused and constricted in the closed channel. So all of that is happening in the central area here. And in the KIR-SUR complex, there's a variety of ligands that also regulate that process. And apparently by preventing or locking these helices into certain conformations. So when ATP binds to the nucleotide binding domain in the channel itself, it blocks the channel. When PIP2, phosphatidylinositol 2, which is a phospholipid head group from the membrane, binds in this region here, it uncouples the inhibition of SUR, so it opens the channel. So you got a couple of ligands having opposite effects on the channel itself, and then you have an opposite situation with the SCR subunit, where when ATP binds to its nucleotide binding domain, it releases its inhibition of the channel. And drugs bind in the center of this transmembrane helical bundle, again, presumably to lock the helices into a particular conformation. And in doing so, they enhance the ability of SUR to block the channel. So these are some of the landmarks that we want to keep in mind. And I would point out that this particular area here, which is at the cytoplasmic domain and near the transmembrane helices of the channel, but also contacting loops from the SUR subunit, is kind of a structural crossroads of the channel. This is where PIP2 binds. And this is also where C166 is. So we suspected that this is the location where palmitylation is affecting that. So we made and optimized homology models of all the KIR family structures. And the reason we do that, we published this in several papers as a minor footnote of how to screen in VLS for dynamics. You know, we believe that homologs can sometimes capture, the structures of homologs can capture the conformation that isn't evident in the structures of a native of the target itself. So we made models of all of the family members and all of the and took the conformations of all the structures of KIR 6.2. In silico, we modified the C166 to the palmitoyl, which is relatively easy to do in ICM with the modify command and the libraries that are there. Then did Monte Carlo plus local minimization of the structure to get a sense of the dynamics of the palmitoyl in all these different structures. And the answer became pretty clear that in one of those structures, there's only one of these structures in the family members that has PIP2 bound. It's been exceptionally difficult to locate PIP2 in these structures. It was actually a controversial structure. It was considered to be in the open conformation because PIP2 was bound, but there was only one PIP2 in the 4 monomers, and there was a mutation made that changed the function of the channel in order to get that crystal structure. So half the field thought it was an artifact, but in this structural screen, essentially, this was the only one where the palmitoyl moiety made a significant contact with another entity in the channel structure. And what it did is it makes a hydrophobic contact with the PIP2. So the way, even though the cysteine is facing away from the pore, the palmitoyl is reaching over from one monomer to the PIP2 binding site in another monomer and presumably stabilizing PIP2. And if you recall, I just said that PIP2 is an opener of the channel. So we proposed that this is the mechanism by which palmitylation opens the channel, is by influencing PIP2. So that gave us also a ligand discovery avenue, and our main purpose was to, again, to use ICM tools to do that. And our main purpose was to test that hypothesis from the modeling of the palmityl oil. And the rationale there would be that in channels, if we found a palmitoyl mimic or a PIP2 mimic, in other words, in that pocket where the palmitylation is and where the PIP2 is, if we find ligands for that pocket, we should see opening of the channel. So in order to do that, as you all know, we've heard some great things about the computational approaches to generating ligands that fit the structure, but the other side of it is you have to have assay development, and that's often a limiting step. We had to develop a cellular assay. It's a thallium flux assay for effluxing an ion thallium that can be detected relatively easily in HEC293 cells, stably expressing Kr6.2 SUR2A. And this is a relatively high throughput assay that can be performed on a flex station. So we use that as our primary screening assay. And again, the choice of primary screening assay is often critical for these types of projects. Only 15 compounds, as you've heard several times today. ICM is pretty remarkable in its ability to identify ligands when the project is set up correctly. And out of 15 ligands, one of them substantially opened the channel. Shown in red here is a compound called pinacetyl, which is a non-specific opener that works on the SUR channel, moiety in the channel. There are no ligands of the known that modulate the function of the K channel itself. And this compound, so pinacetyl is the strongest opener known for these channels, and this compound is equally strong, but none of the others are active, although they have some minimal activity. But the EC50, we did a dose response curve, and it's nicely sigmoidal, although the EC50 is in the low micromolar range, although that's okay for cellular EC50 for drugs. So then another principle is that in the primary screening assay, if you have an active compound, you want to test it in an orthogonal secondary assay. So here we did acellular patch clamp to measure the currents. Glibenclamide is a blocker of the channel, and what we see is that when we infuse the compound, the channel opens, and the channel opening is blocked by glibenclamide. So it's verified by an independent orthogonal assay. And then we did some phenotypic validation of the compound, and this was published, will be published shortly in Frontiers. There's a rare genetic disease called congenital hyperinsulinemia, which you'd want to open the channel to shut off the insulin. And it can be caused by a missense mutation in R301C in the channel. And in these cells, the compound rescues this mutated cell. So this mutation results in a abnormally closed channel, and the compound opens the channel. And then Kr6.2 is also a cardioprotective channel. It opens during hypoxia and ischemia, and in doing so shuts down some of the active metabolism of cardiac myocytes to protect them from those insults and the lack of oxygen. So you can model that with this cardiomyocyte cellular model, and phenylephrine is also cardioprotective in this setting. So what we see is that when we add the compound, without the compound, the cells round up and are not viable. With phenylephrine, which is the positive control, they're preserved to some degree. With phenylephrine plus glibenclamide, which blocks the channel, you get the cell death again, and the compound protects the cells, and they have the same morphology in the presence of hypoxia and reoxygenation. So, but I would point out that KR6.2 in some ways is otherwise not a good target. So even though it can be cardioprotective, it'll block insulin release, which is not a good thing in anything but this genetic condition. So another issue here, I think
and there's 5 minutes left.
Okay, great. I mean, I think we had enough evidence to show to support our hypothesis that the palmitoyl PIP2 pocket is functionally significant. But here we oh, and by the way, the finding of this compound also validated that previous structure where the PIP2 was bound that was controversial. The fact that a compound binding in that same location and screened against that structure indicates that that's a biologically relevant structure. So that's a little side benefit of doing chemically probing of structures. So in terms of selectivity, you also want to have a selective compound. And as I said, these are extremely powerful ion channels in biology, just even in this family. So we tested the compound against KR3 family and the KR2 family. Again, the KR3 family has the cysteine for palmitoylation. And as you can see, by the lack of difference in these curves, the compound is not active against KR3 or KR2. A very close relative of KR6.2 is KR6.1, and the compound is actually selective versus KR6.1. Here, this is pinacetyl. Here's the compound. It actually blocks the channel a little bit. So even though KR6.1 is 70% identical and there's only 2 amino acid side chains differing in the pocket, surprisingly, this compound is very selective for KR6.2, which is both desirable and a little bit mysterious because the pocket is so similar. And then this paper came out after we were characterizing the compound, which explained to some degree this difference, which is that cryo-EM structures of KR6.1 show a displacement of the nucleotide binding domain at the cytoplasmic interface by 6 angstroms away from the phospholipid head groups at the membrane, which is where PIP2 is. And KR6.1 is known to be more resistant at basal state to opening. So we think KR6.1 is designed to be less sensitive to PIP2, and that's why this compound and that pocket is not as active. But it's more because of the domain structure of this multi-domain protein than the slight differences in the pocket. I mean, we know that even slight differences in the pocket can be targeted by chemical groups, but in this case, there's another explanation. So conclusions, the KATP channels are regulated by S-palmitoylation. That's a new finding. We propose this mechanism that the palmitoyl stabilizes a ligand. So that's a mechanism that hadn't been thought of before. And that identifies a novel druggable pocket central to the mechanism. And the VLS targeting this pocket efficiently identified at least one compound that's a channel opener. This is the first and only KIR subfamily specific opener identified to date. So it shows the power of following the mechanistic clues in the structure to drug so-called undruggable targets. So as the title of my talk points out, the story emphasizes something that my lab is thinking about, which is VLS for mechanistic chemical probes first, and then ultra large VLS, the giga VLS for the drug development. The reason that's important is so I'd be interested in hearing from the meeting this week. We'd be interested in a minimally sized, maximally diverse subset of the giga to do quick searches. So if you can do billions of compounds in a couple of days, why not do a few million in less than an hour and get the probe, and then start testing that and run the giga in parallel. You know, the rate limiting step, what we'd really love to speed up is how long it takes to order the compound and receive it. And it'd be nice to be able to do the whole thing in one day, talking about unrealistic or idealistic things. But that would go a long way towards accelerating some of these projects. So this pocket may be functionally sensitive in a variety of ways. And this is another story, and we've heard about it from Nadia and others, that we may not be able to talk about how challenging it is to drug undruggable targets for much longer, because there seem to be lots of ways to do that. So I believe most targets are druggable. I also believe that you need to have a pocket. Maybe the shallow grooves aren't good enough, but usually you can find one in almost everything. This should translate to the KIR3 and the rest of the KIR6 family because they are PubMed related. I didn't talk much about the slide helix, but it's the key helix near this pocket that connects the SUR, the transmembrane helix of the KIR, and the PIP2. And so any pocket near this key mechanistic keystone, I think, can be a target in these channels. I think there's some lessons about selectivity. This was very selective in the KIR family, but surprisingly, I didn't show this, but we found it might cross-react with a sodium channel. So this mechanism may be more general, and that creates promiscuity. But we did find that the selectivity might derive here from both the pocket shape and the broader mechanism. So when you think about what Ruben said at the very beginning, like we're interested in multi-target profiles, he was, I think, focused on kinases, but ion channels also, we might need multi-target profiles for drugs. It's a major drug target. We might want to be thinking about a combination of both pocket promiscuity and mechanism promiscuity, so that you need to have a unique pocket, but the pocket also has to avoid some surrounding features that have to do with the mechanism that may be common in many other targets in the same class. So there's a lot of people contributed to this. Will Nelly was one of the authors, was on the original polymethylation paper, and Molsoft, of course, for not only the tools, but the inspiration for this kind of work. Thanks.
Tim, thank you very much for that talk. Thank you. It's very interesting.